And if a trait disappeared, it was gone forever. So that's sort of what we're trying to say here, where the characteristics of both parents irreversibly or blended irreversibly in their offspring. It'll never show up again. We want to actually look at this one and disprove this idea this morning with his experiments on pea plants. Now, when he studied pea plants, he looked at 7 different characteristics. So there's some vocabulary you want to get a little bit used to. The word characteristics is the same as the word trait. And that is the thing about the organism. Like, I have a trait of silver hair. Some of you have a trait for red hair, for blonde hair. Your skin color is a trait. Your resistance to disease is a trait. Food allergies is a trait. Allergies overall is a trait. Well, he studied the traits or the characteristics of 7 different things. One was the color of flowers in the pea plant. The other was the position of the flower on the plant, the color of the seeds, the shape of the seeds, the shape of the pod, the color of the pod, and even how tall the plants were. We're going to focus on his first one, which is the actual color of the flowers. Now, he's trying to figure out how organisms pass on these traits, these characteristics to the next generation. He used pea plants because they're cheap. They're easy to grow, and you can do them over and over and over and over again. Well, what he wanted to do is first he wanted to kind of, in a sense, purify his population. He wanted to create what are called true breeding flowers. You guys might think of true breeding as a purebred. Right? You got purebred for dogs and for horses and such. Meaning basically that when you make that animal, you produce high quality stock in the next generation. He wanted to produce true breeding, which means that anytime you cross that flower, it'll always produce the same thing. So he wanted to create pure breeding purple flowers and pure breeding white flowers. Meaning that anytime you bred white flowers together, you always get white flowers. Anytime you breed purple flowers together, you only get purple flowers. This was a painstaking process for him. It would test your patience. And this is what he did. He took a garden full of pea plants and he planted them, and a garden full of pea plants and he planted them. When the plants produce flowers, he says, okay, this is my dedicated purple plant. These are my dedicated white flower plants. So any white flowers I find in my purple bed, I'm going to pull them right out and throw them away. Any purple flowers I find in the designated white bed, I pull out and throw away the purple flowers. Then I take my remaining purple flowers and I take a little paintbrush and I pollinate every single flower in my bed. And he's doing thousands of plants. Over here I do the same thing. I take a different little paintbrush and I pollinate all of the white flowers in here. They produce seeds. I gather up all the seeds.
This is the monk, right? Like the
Yes.
monk did in the monastery?
This is monks, yes. This is what Gregor Mendel did. Then when all these plants die, and all those plants die, I take their seeds that I got from here and I plant them back in the same pot. I plant all these seeds in this pot. I let them grow back up. Well, I've got mainly purple, but I still got some white ones in here. So I throw away all the white flowers. I pollinate all the purple ones again. I've got some purple ones in my predominantly white flower bed, so I throw away the purple ones and I pollinate the white ones. They each produce seeds. These flowers die, I take their seeds and I plant them back in this one. And I do the same thing here. The white ones die, I plant their seeds. And this takes a few years to do. But I've gotten to the point finally now where whenever I pollinate my purple flowers amongst purple flowers, I will bet my soul that all I'm going to get are purple flowers. And the same over here, that if I pollinate the white ones, all I will ever get are white flowers. I now have true breeding flowers. Now, of course, there will be the odd mutation perhaps, but we're not looking at odd mutations. Now what he has is a pure population that if you were to say, John, I want a purple flower, I'm going to sell you the seeds and I will guarantee you get purple flowers. Same thing over here, but now for the white ones. Now he takes those pure breeding strains and he creates what's called a hybrid. He mixes them. He pollinates them together. Now, here's where Mendel's day comes in handy. In his day, people thought when different individuals mated, you got a blending of their traits. So instead of being a nice, good, deep purple, it should be a much fainter color. Sort of like mixing red paint and white paint, you should get pink. Well, what he found was they're all purple, and the same color as the parents. Well, we refer to this as the F1 generation. And the F stands for filial. And we just talk about F1s and F2s. Filial. And F1 is easier. All right. Now also notice that they're all purple. That also matches the other thought in Gregor Mendel's time, that you now will lose a trait. You've lost the white trait. Gone forever, never going to be seen again. He had enough hindsight to actually, well, what would happen if I bred these guys amongst themselves? So now I take that first generation that I got from blending these two, and I create a different bed of flowers. And I plant all of them out to see what happens. Well, when he got the seeds, he planted them. And what he found was, in the next generation, when he planted those seeds over here, that every time he did that, out of every 4 flowers, one of them would be white. And that was shocking, because if the idea was true that you lose a trait, well then where did this come from? And he had enough math to realize that there was a ratio going on here. For every 3, I get a 1. So it's called a 3-to-1 ratio. So whenever he would plant these, 75% always came to be purple, and 1/4 always came out to be white. And so now he's beginning to look at, all right, well, how can I explain this? And there's some words that he kind of created that we're going to be looking at. The idea of dominant versus recessive, more specifically. So we can look at the F1 and F2 as, the first generation and second generations. And then the word monohybrid. Mono means one, because we're only looking at one trait, the color of the flowers. If you were to be looking at 2 different traits, like the color of the flower and the position of the flower, that would be called a dihybrid, because you're looking at 2 different traits at the same time. If you were to examine 3 traits, that would be a trihybrid cross. So monohybrids are really easy. You guys did them in high school probably. Maybe you didn't remember it or didn't realize that's what you were doing. So let's look and see how he does this. He did show that there was no blending of traits, because if there were, my purple flower should be a light purple, not the same purple as the adults or as the parental strand. And he called it a heritable factor. He didn't have the word gene just yet in his mind. But it didn't disappear, whatever that thing is. Yeah, it disappeared in the first generation, but it did reappear later on. After a couple of years, he said, all right, we need to think about these as being genes, and that I'm getting a different gene from each parent. Now, if we're looking at the trait of flower color, that means I have a gene for flower color. But if the male plant has, let's say, the purple gene, and the female plant has the white gene, and the genes for flower color, then we use the word allele for the different version of the flower color gene. I have a purple gene and I have a white gene. So we have alleles. They're simply just versions of the gene.
Alleles?
Well, then he created these ideas of dominant versus recessive. Now, he had the idea that means each parent, each plant must be contributing one allele to the next generation. I'm pollinating 2 plants, so I get 2 alleles to create that next generation for the trait of flower color. Now, the word dominant means that those genes produce more of the protein than the other version, the other genes. So in a sense, a dominant trait is a dominant gene that overpowers the production rate. So we're going to have 2 genes here. We have 2 alleles. Let's say that the gene is for cookie production. We're going to make cookies. This one makes 25 cookies. This gene only produces 12 cookies. This one is more dominant than this one. They're both producing cookies, but this makes more of it. And so we tend to see the more dominant trait. That's what we see physically. So don't think of it as producing nothing. It just doesn't produce as much as the dominant trait will be producing. So it's like dominating, like someone dominates over you. Well, let's see how this works in terms of his pea plant experiment. If we kind of take a step back and look at yesterday for a minute. So we have 2 chromosomes. Let's make one from mom and one from dad up here. Now we know they're the same chromosomes, right? And when we mean the same, what we're trying to talk about is that each of these 2 chromosomes have the same genes. There's the P gene, the A gene, and the B gene. Yes, they're from maybe 2 different individuals, but we're lining them up by their same chromosomes, like you do in a karyotype, right? You've got 2 in the number 1, 2 in the number 2s, right? One of those came from mom, and one of them came from dad. Well, there's a little more vocabulary we need to go through today. So if these are homologous chromosomes, because they have the same genes, but from different people, we can also use the words homozygous and heterozygous to describe the alleles of those genes. So here's the character, the trait for the P. There's my P gene. Since both chromosomes have the same capital P, we call them homozygous because they have the same allele. They have the same version. Over here, on a different trait, we now have 2 lowercase a's. Well, that means they have the same version of the gene. They both have a little a. So they have the same alleles. Again, they're homozygous. But we use a lowercase letter to designate or to represent a recessive trait. We use a capital letter to demonstrate a dominant trait. So since both have a dominant trait, they both have the capital P, they're homozygous for the dominant allele. These individuals each have a recessive allele, but they each have the same version of that recessive allele, so they're homozygous recessive. Now, all you need is one dominant allele for the trait to be seen with your eyes. So when I think about Mendel's experiments, and he said, I got all purple flowers, I realize there's 2 ways that can happen. Those all purple flowers in that next generation, they only have to have one dominant gene. They could have a recessive one, but the dominant one is dominating over the recessive, so they'll still produce the purple color. Now over here, we have the same trait, right? But we have 2 different alleles for the same trait. There's a dominant allele and a recessive allele. Hence we use the term heterozygous. You don't need to use the word dominant or the word recessive, because hetero means you've got one of each. And we know the rule. If you have a dominant allele, you will have the dominant trait. Kelly?
Does this mean that even with his true bred flowers, they weren't true
No.
bred?
And we'll see why that's true when we do them. So I will, let's say, quote, for all intents and purposes, guarantee that he had homozygous dominant plants. Except for the odd possible mutation that would happen. All right? And we're going to work through that so you can actually prove and see that's why he knows he had homozygous dominance. So are we okay with these terms for a minute? All right, now a little self-pop quiz. Don't panic. But what I want you guys to do is I want you to look at this slide that we just talked about. I want you on some scratch paper, or as I provided in PowerPoint notes, on the next slide, I want you to draw me what these would look like in metaphase 1. What would those look like? You can ignore crossover events. Just show me what they would look like in metaphase 1. And we'll see who's comfortable doing this and who's like, I haven't got a freaking clue just yet, I'm not there. Just give it your best shot. Not for a grade. what are you gonna find in metaphase 1?
I don't know. I think I know. Then pink.
Now, you're gonna wanna get good at this, because I'll guarantee you, you're gonna be at something like this on your next test. Once you get the hang of it, it'll take you less than 30 seconds to do. It just takes practice. It's a new skill you're trying to use. It takes time to learn how to do that skill. Red and blue. All right. So let me show you what I would have put. First I draw a circle, then I draw like an equatorial plane. I know I'm going to have centrosomes involved, so my chromosomes can either go this way or they can go that way. I'm so used to doing it this way, I'm comfortable with it. I'm going to stick my centrosomes down here. So now I know that I'm going to be spreading this way. All right, well I've got a red one, so I'm going to quickly show a red one here. And I know it has to go through the synthesis phase. I need to make a copy of that chromosome. So I have to show its sister chromatid. So I'll draw another one here. That's a bit too long. Have a bulge there. And then I'll draw its other one like right here. Well, that's the mom's version now replicated. Now I need to do the dad's. So then I'll have a little bulb here and a long thing there. It has to go through replication. If I come around and go like this, I can draw my centromere if I want to right there. I can draw it like there. And that's what I would look at in metaphase 1. What word can I use to describe this thing, this mass of stuff I see in the middle? How can we refer to these things? The tetrad or what? Homologous pair. Because what does homologous mean? They have the same genes. And I have a pair of them. So it's a homologous pair. It's a tetrad because we've got 1, 2, 3, 4 sister chromatids involved. Now, is this the only way I could have drawn it, or could I have drawn it another way?
I thought you could.
So could I put the blue one on top, Kelly, and the red one down here?
It could rearrange either way.
And that's the idea of independent assortment, right?
Yeah.
How they line up is just by pure doggone chance.
Yeah.
But you need to show your homologous pairs stacked. I can't draw the blue one over here, right? Because then they're both going to go to the same cell, and now you already have a non-disjunction event. So I have to stack them up so that the idea is that when these guys divide, this cell will get 2 reds, and this one will get 2 blues.
They divide because the spindle is pulling them apart.
Right, because during the anaphase part, exactly.
Can't be independent.
Huh?
Can't be can't be parallel like this way.
No, it can't be paired. They have to be stacked. Because one has to go to one cell and the other one has to go to the opposite cell. If you have them side by side, right, we don't do this. It's everybody that's in the middle will go up, everybody below the middle goes down.
Yeah.
So in metaphase, they're actually connected to.
That's what I thought.
Right? Like due to
Who's the they?
The homologous pair is.
So you're saying that all 4 are connected together?
So the crossover point, the point at which they're connected through crossover,
Right.
right?
That's
They're
what
attached to one another in some way before being pulled apart at the end of it.
For a moment, yes. So that's why I said ignore crossover events, right? So for a moment they're connected, but once they're done crossing over, they're no longer connected.
Yeah, right.
So then this will go that way and these will go this way. But in terms of what you're thinking about, I could have drawn it kind of like this.
Yeah, I was gonna say that.
Right? That's why I said they'll ignore crossover. So you'd have this idea.
Right.
All right, but they're no longer connected. All right, so
Okay.
let's look at how we can be comfortable in
So I had a
proving
good idea. and
that
it didn't-
Mendel was
okay,
right
I
in
don't
terms
do it
of the idea of having
Okay.
pure breeding plants.
This is actually gonna be.
So this thing here is called the Punnett square, and you may have done this when you guys did high school biology, and you may not have ever been exposed to it. It's not complicated at all. What you do first is we get rid of our cell here. And the first thing you do is you draw a box. That's pretty easy to do. So I draw a box. And then divide into 4 pieces, because I'm going to get 4 offspring. And then in terms of what gets put where, we put gametes up here, and put gametes over here. So I can do columns and rows. So I'm going to put gametes over here as well. Now, gametes are what you guys look at the end of all of your meiosis. So if you remember, in terms of the stages of meiosis,
Gametes.
where we started off with a single cell, and then it's going to go through the first round of meiosis, so it creates 2 cells. So there's meiosis 1, but then it has to, each one of these has to go through meiosis 2, and so they're going to produce 2 cells each.
Right.
All right, so I start with 1 and I get 4 when I'm all done. We started off with 46 chromosomes, but we're going to get 23 in each one by the time we are all done. If we look at a little out of space, we'll do it down here. Some vocabulary that we need to talk about for the rest of this is to do it this way. If I have a cell that has green. No, can't do both green. That would be a genetic problem. Use black. There we go. And then we'll put, a green one over here. So I've got 1, 2, 3, 4 chromosomes. The size means they are homologous. So I've got a mom and a dad, a mom and a dad.
Same.
I've got 2 of each chromosomes. Over here, I've only got 1 big one and 1 small one. So if you want, these could be your 5 beads and your 3 beads from yesterday. So I've got 2 5s and 2 3s, 1 5 and 1 3. Since you have 2 of each kind, that makes you diploid. So that would be then a diploid cell, because you've got 2 of each kind, 1 from mom and 1 from dad. Here I've only got either the mom or the dad for the long, and either the mom or the dad for the short. So this would be haploid. What are these guys? Are these diploids or haploids at the end of meiosis? They're all haploid, right? You only have half the number of chromosomes. You don't have mom and dad. You have a mom or a dad version
Right.
of all of those chromosomes.
Okay.
So all of these are going to be haploid. Well, that's what our gametes are. They are these cells. You know, if you want to just do this, right, make little sperm cells out of them. Those are the sperms. All sperms are haploid. All ova are haploid. And when you add a haploid to a haploid, you make a diploid. Well, what you want to do now is figure out the probability, the chance of what your offspring will get, depending upon the different possible combinations of the gametes donating their alleles. So if capital letters are predominant, and we're doing the idea of purple, I first have to create a legend. So I'm going to say that capital P equals purple. I then am going to say that lowercase p equals white. You're first creating a legend for the code you're going to use in a Punnett square. Now there's a problem with some letters of the alphabet, right? Capital P and lowercase p, I can't tell them apart sometimes, right? So you need to put an underline under it to designate that it's not a capital letter. A's and B's are easy because each of the letters looks different, right? A capital B is different than a lowercase b. But think about an S, right? You got to do a little bit differently. Now here's the other key thing about genetics. I picked to use a capital P for purple because purple begins with a P. That's why I used a P, right? You need to use the same letters for both the dominant and recessive. The only difference is it a capital letter or a lowercase. You can't go a P and a W. Different letters mean different traits. So now I've created my legend that capital P's are for the dominant trait and lowercase are for the recessive trait. We are saying that this plant, right, is the female. So we're going to give her a lowercase p and a lowercase p. The data could go up here. It doesn't matter where the sex goes, all right? Over here then, we put down the capital P's are over here, and that means it's a purple plant, and there's a capital P. So now I'm saying that these are the gametes that'll come from the female, and these are the gametes, or the alleles, we should say, right, for the color of flower petals. Capitals and recessives. Now the way I do it to keep straight is I do the capital letters first. Because the grammar of this is capital letters always come first. So I'm going to go across this way and take care of my capital letters first. Capital P, that row's done. Capital P, that row is done. I've now done my capital P's. I then come down and do the recessives down the columns. Lowercase letters always come second. So now I'm showing the combination of alleles that would come from the male and female after fertilization events. These then are the offspring. So these are the offspring. And this also represents what's called the genotype. Right? The type of genes that are in the individual.
Genotype.
This over here, purple and white, are the descriptions of the colors the petals have. This is what you guys see. This is because of the genotype. So this we call then the phenotype. Which is the expression of the genes, which we'll walk through a bit.
So capital C and then the one called offspring. And
So
this one
Yes. All right, so now look at the slide we talked about in terms of the combination of alleles. Is this a homozygous or a heterozygous individual? It's hetero because I've got one dominant, I've got one recessive. Capital letters designate the dominant trait. You only need one dominant to have that trait. You only need one dominant to have that phenotype. So all of these are purple. That's because I used a purple flower to produce the next generation. And every time I do this, at least with this combination, I will always get purple flowers. Because I took a pure breeding white with a pure breeding purple, and my F1 was all purples. Now you can see, as learning genetic students, that I can see the allele for whiteness. It's there. It hasn't gone away. It's just being overpowered by the allele for purple. And it's just a protein pigment. All it means is the plant's making way more of that pigment than this one. So all I see is the purpleness. Now,
I have a question. So, said purple and white is gonna be more purple, but there is still white. But how about different colors?
The what?
Instead of white, if it's black.
That's a different trait. That's a different gene. That would be a whole different scenario. Okay, so this is when you have purple and whites together. So for here, there are two ways you can get purple. What you want to do is you want to list all the different genotypes that you would get from your cross. This one cross is really boring because all we get is capital P, lowercase p's. All we're going to get is the genotype big P, little p. All we're going to see are white flowers. But what if we change it up and have you guys quickly work out this one? I'll do that one over here. And again, you'll get some practice in how fast you should be able to do this on your next test. It should take you about 40 seconds to do, once you get really good at it. So first you make your box, you divide it into 4, that's a monohybrid cross. You show the gametes you're using for that cross, and then you simply plug in what you've got. So don't look what I'm doing, see if what you do matches what I just did.
B.
Okay,
Might have did that on accident too.
now. Right, this is what you should have produced. Now, the more you start doing this, the more you start seeing a pattern in genetics. I will guarantee you that every time you do a monohybrid cross with a homozygous dominant and a homozygous recessive, you will always get this result. Every single time. It becomes a reflex to you in genetics. This is what's known as a monohybrid cross between two heterozygous individuals. You will always get this pattern. Every single time. Now, everybody get this done? Everybody got the same letters in the boxes? All right, so now we want to list all the different genotypes that were possible. So what you say is, all right, I always start up in the first box and say, well, I got a big B. All right, there's that. So I cross it off my list. Any other big B's? Nope, so I've handled that one. Let me go over to this box. It has a big B, little b. So I write that down, big B, little b. I've done that. Anybody else have it? Yeah, this one does. Okay. But I've already written it down as a list, so it's not different, but I've already accounted for it. So I'm done with this one. These three have already been handled. This one's my last one, has a little b to it. Cross it off. I've now accounted for all the different gene types that could have come from a breeding between this heterozygous dominant individual, in a sense, right, and this heterozygous individual. But then I want to talk about the odds, the chance, the probability of getting this combination of alleles from a mom and a dad, and this combination, and this one. So I go back and say, well, how many boxes do I have? I've got 4.
4.
Of my 4 boxes, how many had a big B combination?
1 out of 4.
1. So you had a 1/4 chance. Or 25%. Now, so I've accounted for that one. How many of my boxes had a big B, little b?
2 out of 4.
You go 2. 2 out of 4,
It's
right?
50%.
2/4, or reduce that to 1/2. I've accounted for that one. The last one is little b. How many of them have it? 1 out of 4, so I have a 1/4 chance of getting that genotype.
Don't go into math statistics now.
These are chances.
Oh gosh.
And they're odds. They're not guarantees. So you could have 4 children, or 4 and all they have are little b's. It's just the chance of having it. And we'll look at what happens with future generations. Now these are the genotypes. I can also then, if I designate what big B stands for, like black, and little b for brown, then these two would be black, and these would be brown. Because you have to have both recessives to have the trait. Remember, all you need is one dominant to have that trait. Well, let's do it one more time. Let's take a look at now, we cross these two, homozygous recessive, homozygous dominant. Everybody was heterozygous, which could only be dominant traits. Let's now cross two individuals of this to see what they would get in their next generation, or F2s. So we come over here, and I'm just going to get rid of the gametes inside, keep the box, because the words still apply. Now we're going to do heterozygous dominant. Still follow the same basic rules. I'll do it that way in colors. I'm going to do the capital or the dominant letters first. So I'm going to come down here. You don't always have to go this way first. I'm going to come down this way instead. So there's that P and this one. Then I come over here. That would be a lowercase p. Come down here, a lowercase p. Lowercase letters always come second. Then I come over and handle this. So there's a big P there, a big P here. See, it went first. This one goes behind the capital P, and the only one place to put it. And I go, that's different. All right, so now I'm going to list all my genotypes that I got from this cross. We're going to have a big P, that handles that one.
So-
I'm going to have then a big P, little p.
So it's the same as over there.
So I've got this. Yeah, it would be. That means I've got two of them, so I've handled those two now. And I've got this one. That's a lowercase, lowercase p. So you guys tell me, what color is this individual going to be? This one's purple. What color is this one going to be?
Purple.
Purple, because it only needs one dominant allele, right?
Yep.
So that's purple,
White.
and these are white. Now, of my boxes, how many total boxes are purple?
4. I mean, no, I didn't mean to say that. 3.
We got 3 that are purple, right? Purple, purple, purple. 1, 2, 3 out of my 4. So 3 of these, and how many are white?
1.
We got 1. A 3-to-1 ratio. 3 purples for each white.
3 to 1.
That's what Mendel got when he did his pea plants. Every time he crossed the second generation, he got 3 purples and 1 white. Now he knows these are going to be homozygous recessive. He does not know which of these are going to be homozygous dominant or heterozygous dominant.
Right.
Because you can't tell by looking at them. So that's one of the rules I want to make sure you guys get in our minds, is that if I can see the phenotype, I don't know with certainty what genotype they have. I can guess. Because to be purple, right, I could be a big P, or I could be a big P, little p. There's an or in here. There's only one way to be white, so I know what that one is.
The lowercase.
But if you tell me the genotype, I will guarantee you the phenotype. Because I know that this produces purple. I know that produces white, and I know that produces purple. So knowing the genotype will tell you for certainty what the phenotype is. Knowing the phenotype, there's a possibility. There are this or that or this in terms of combinations. So far okay? Okay.
So phenotype's the expression of the genes?
It's the expression of the genes, exactly. All right, now that expression can also just be physiology, like your ability to tolerate alcohol, or your inability to sneeze when you get a bunch of pollen, or your ability to sneeze because of it. All right, so now one last little teeny thing is we're going to work on these in lab after we do our next gen plating technique.
I know traits that are not like one or the other, but it's like,
Oh yes, we're going to walk through all those. So we're going to be looking at sex-linked traits, complete dominance, incomplete dominance, and codominance, the major categories of dominance tomorrow in lab. So you're exactly correct. There could be scenarios where if I see a big P, a little p, it actually could be a blending effect actually. Whereas big P's are one color and little P's are another. You get 3 different colors from 2 parents. And we'll teach you how to recognize that.
The independence.
All right,
but not
so what we did
happening
over
at the
here
same
is
time.
chances.
You know,
But I want to also know the idea of what are the odds of getting, let's say, a child that's both homozygous dominant and one that's heterozygous dominant. Or I could say, what are the odds of getting a homozygous dominant or a heterozygous? In other words, ands and ors. So think about a coin toss for a second. If I toss it, normally only one of 2 possible events will happen, unless you have a trick coin that lands on its edge. So we're not going to consider that. Then I toss a two-headed coin, that's what's going to work well. If I toss a normal coin, right, what are the odds of it becoming a head?
50%.
1 out of 2, right? Because there's one side's head, one side's tail. So here's the question. What are the odds of tossing a head the first time? It's what?
2 out of 4. I mean, 1 out of 2.
2 out of 4 is only a 2-sided coin.
I know.
You're using a funky coin.
I know. I'm just
Right?
mixing up stuff in my head. I was looking at this chart right here for some reason. I don't know why.
All right, so the odds of getting a head
I wasn't paying attention either. Sorry.
versus the odds of getting a tail. Well, it's a half chance for a head and a half chance for a tail.
50-50.
So now, what are the odds of me tossing the first and getting a head? And what are the odds of tossing and getting a head the second time?
1-4.
Well, just individually. Just the first toss is a head. Well, what are the chances of getting a head the second time?
50-50.
What are the odds of getting a head on the 55th toss? It's still 1 and 2, right? Because the previous toss does not determine
The second one.
the other upcoming tosses. All right, but now,
The events are independent.
what if I said that the first, let's do it this way, the first equals a head and the second equals a head? Well, you take the odds of each individual event, the odds of a head is a half, and the odds of another head is a half, but I'm using the word and in my thought process. The toss of getting a head on the first and getting a head on the second toss. You're going to multiply those 2 separate independent probabilities. So the odds of tossing a coin twice and each time getting the head is 1/4. That's
Yep.
1/2 times 1/2. If you were to say, what are the odds on the first toss of getting the head or on the first toss getting a tail, you take the odds of each independent event, we have a half chance of getting the head, you have a half chance of getting the tail, but now my magic word is the word or. Then you add the individual probabilities. So you're going to add them. Which kind of makes sense because if I say I toss a coin, what are the odds of it being a head or a tail?
50%.
Well, duh. It's going to be one or the other for sure. You're guaranteed to get that to happen.
Comes 2, 4.
When you do probabilities, they must always add up to 1 when you're completely done.
Yeah.
And we're going to be looking at a di-hybrid cross, not today, but we got 2 traits instead of just 1. And listing all the genotypes and all the phenotypes and all the probabilities of getting all these different genotypes that come out. Does this make sense?
Yeah.
All right, when to multiply and when to add? All right, so then you come back and look at, this makes a lot more sense when we do a di-hybrid cross. But it's the same thing we're doing over here, right? What are the odds of getting this genotype? Well, it's 1 out of 4 and 1 out of 4. So the odds of getting this one or that one, you add the 2 together, which gives you overall 1/2 chance. All right, that's all we needed to do for this. So you're done. All right. Keep in mind that this is the big idea happening today. This is the focus of a lot of people's thoughts and attention. especially if you're in Las Vegas, because of the Cubs, right? They're saying, what are the odds of winning 3